Thank you.
Thank you so much, Walter. And thank you so much, Susan. That was an amazing story. And we're so grateful to you for having shared with us today. I am going to kind of immediately segue us into our next session. So this is a session on clinical solutions following COVID, new opportunities and new times. I do want to briefly just introduce that we have the moderator, Geraldine Bliss, is going to help carry us through the question and answer after we see a broadcast of four presentations from Liz Barry Kravis, Ray Dorsey, Laura Lee Johnson, and Ann Pariser. So without further ado, I'm going to ask our logistics contractors to start the broadcast, and then we will all come back live for your questions and answers. Once again, please feel free to use the hand raise feature or type in the chat.
I want to thank the organizers for inviting me to come and talk on this panel about COVID-19. And I'm going to focus on COVID-19 and clinical trials. And I work as a pediatric neurologist at Rush University Medical Center. And as the Washington Post pointed out, our hospital was built for a pandemic. So we had to be ready to make our clinical trials conform to the pandemic and live up to our reputation. So there were a number of things that we had to do in March of 2020 when all of a sudden out of the blue, we were hit with COVID and we were running like 20 trials, and these had all taken a great deal of time and effort to set up, and we did not want to lose all the data. So we had to do a number of things rather rapidly because patients couldn't come to our site and we couldn't interact with them normally. We had to create remote visits by video conferencing. We had to figure out how to get labs and EKGs from people remotely. We had to mail drug. We had to do IRB and FDA submissions and notifications and do essential visits, figure out how we were going to actually do essential visits on site and how are we going to do neuropsychological testing for assessments in the in the time of COVID. I'm going to talk a little bit about each of these things, and we put these things all in place and what are their advantages and disadvantages that we discovered rather rapidly because we were forced to in the COVID environment. So video conferencing at home for visits. There are some great advantages to this in that you would see the patient in their natural environment. They were less stressed from travel and the parent was more free to converse because they could just have the kid go play in the home. The disadvantages of this are there's simply parts of the exam we cannot do. The patient often didn't really want to interact with the screen because they didn't view it as a person. And so you couldn't interact with the child as you would in clinic. And we needed a way of getting medical measures like labs that you needed the person physically present for. So we did have processes that were put in place in various trials for getting vitals, labs, EKGs, EEGs in the home. And some of this was where the patient went to a local lab, but in this case, they still had to go in somewhere and there was a potential COVID exposure. And then there were insurance billing issues because the family would have to pay up front and then get reimbursed from the study. And then what many companies did is to hire a home nurse or a testing company that did the tests at home and then sent these to a central lab or reader. This was less risk of COVID exposure, but the test quality was more variable because there's quite a bit more variability with this process than there is in a lab. Drug mailing and bottle return. This turned out to be kind of a nightmare for multiple studies. We had to send drug to patients and we had to get it back somehow and count it all. So sometimes there would be an out-of-site pharmacy where we could mail the drug directly from the company. Sometimes we had issues with delivery. The couriers were very variable and critical in order to make sure the drug arrived on time. And then we would have to count a lot of bottles the next time the patient came in. And at the end of the study, we would have to have patients mail back stuff for drug accountability. So this would involve a lot of pestering of families. IRB and FDA submissions and notifications were really not too much of a problem during this time. Both the IRB and the FDA were generally very accepting of protocol changes and deviations for COVID. And we were allowed to adjust protocols in the moment as needed and then submit later to the IRB for blessings. And the FDA submissions were mostly information about what we had to do to change a protocol to make adjustments for COVID. And in fact, they were so accepting of COVID that every time we had a deviation, we were thinking, how can we make this due to COVID? So we did have to do some on-site essential visits during the height of COVID. And this was when safety monitoring or key assessments could not be done remotely. we would need to do an infusion. And so we actually needed the patient to put something into them by IV or intrathecal infusion. And then we had issues around lack of anesthesia certain weeks in neurodevelopmental patients because anesthesia was not active every week of COVID. During these visits, we used full COVID precautions, masks when possible, although kids with neurodevelopmental disabilities don't always keep the mask on and are sometimes disturbed and want to pull the adults' masks off. And then we lived in a sea of disinfectant where we just disinfected everything constantly. And then we had to get exemptions in some cases to have both parents present when the patient was too hard to handle for one person. And sometimes we could not get the patient here because of COVID restrictions or quarantine. And this is just a little trip we went on to Oklahoma for a key study visit in a trial that were data that we did not want to lose. So we went and tested the patient at home. There were neuropsychological testing issues in that the test was not valid when the examiner was masked in some cases. And this varied a lot between tests, and we had to make educated guesses about which items and tests would be valid. So we did design substitute testing in some cases for items that were not valid. Clear masks were not really a solution for this. And in some cases with COVID testing of everyone, we just did testing with the examiner unmasked. So I'm just going to go through an example of the trial adjustments we made for one trial, our NeuroNext trial of the effects of AFQ-056 and mGlu blocker on language learning in young children with Fragile X syndrome called FX-Learn. And this is just one example out of like 20 trials we had to change. So you can see how much work this wound up being very rapidly. And this is an NIH-funded trial run through the NeuroNext network. I'm the PI, and it shows an example of the many processes that we had to implement to keep a trial running and not lose all the data we had worked for years to set up to collect. So this is basically the process. We had to survive the first onset, stay ahead of it, and get everyone working in unison at all the sites, and then grab the moment and take things to a new level. So the advantages we had in FX-Learn is we were already doing a remote language intervention. So we had some remote processes set up. All assessments to that were already remote, related to that were already remote, and families had Bluetooth and computers for the remote language intervention. We had already made 2 visits remote because there was just no safety signal and to decrease travel burden. And we already had a remote process for calls for dose adjustments and drug dilution. So we already had some remote processes that we just built on. And what we did, this is the study, it's a long 20-month study because we're studying learning. And so there were 3 visits we deemed absolutely essential to be able to interpret the study. And then 2 other visits we thought were pretty essential. And so we tried to do those in person. We had already made these 2 blue visits remote. And what we did in March of 2020 is make these other 6 visits into a remote process. We're doing individual site management until we came out with a process shown down here, guidelines and procedures document for COVID-19. By the end of March, we were one of the first trials to get this out. We created a visit hierarchy, like I showed you, for what visits would have to be done in person and what would not. We created a process for the remote visit, what we were to collect, how to mail the drug, how to do the forms and assessments. We made database modifications. We extended the visit window for essential visits to make sure we got them in person, but kept them within reasonable ranges to get valid study data. And we allowed sites to do their essential visit at rush if the site could not see the patient. So thus far, we've conducted almost all visits, either remotely or in person, based on the requirements. One of our issues was our primary outcome, which was being done at many visits, was an in-person assessment, a play test of communication. So you can see it's at a lot of visits, and we would be missing a lot of these from the remote visits. So what we did was we modified this. It's a play test that the speech therapist gives various toys to the patient, and then we measure the patient's communication. So we modified this to make it a remote assessment where the parent acted as the tester and the SLP Bluetooth what exactly for the parent to do. And the parent acted as the arms of the SLP, and then we videotaped it. We shipped toys to the families to do the standardized assessments. And this has actually worked surprisingly well. And currently, most of the weighted communication measures that we've recovered remotely have been adequate for for analysis. And so this is a new method of remote assessment that we actually created during the study. Some of our toys turned out to be things that don't work well in COVID, like they have bubbles or people produce spit while they're using them. So we had to substitute toys, and we had a bubble gun and a balloon pump and a rocket launcher that we added to our repertoire, and these were considered valid substitutes. We trained all the remote weighted communication assessors. And then our speech therapist actually took us beyond NN 107. It turned out our central SLP team that was delivering the speech intervention remotely was doing such a good job that they were recruited by multiple states to give talks to therapists and early childhood educators on how to do remote language intervention when all the state early intervention programs had to go to remote delivery. And so we consented families for use of intervention videos and created web-based training modules. And then these SLPs talked at virtual conferences. So we really made our contribution through this study to the adjustments that had to be made for COVID for kids with neurodevelopmental disabilities. And I think the challenges that we addressed during this study are going to bring new approaches going forward. This is a huge effort, but we now know how to carry out all these processes, and they can be carried over to new trials. And new trials are now actually being designed with more remote assessments and visits to ease participation burden particularly for rare disease populations that often have to travel to study sites. So that's my summary, and I'm going to end there.
Greetings, everyone. Delighted to be with you today. I want to talk to you a little bit about the future of clinical trials, especially after coming out of the COVID-19 pandemic, what lessons we've learned, and how clinical trials are likely to change going forward. First, I want to start with that by looking at how clinical care dramatically changed in the setting of the pandemic, and then talk about the transformation of clinical trials. So before COVID-19 happened, telemedicine was a niche industry. Medicare spent less than $1 out of every $1,000 on telemedicine, but that changed rapidly in the span of just 4 weeks. Medicare visits by Medicare beneficiaries via telemedicine went from almost non-existent to 1.7 million visits a week. So you went from something that almost didn't exist among Medicare beneficiaries to 1.7 million visits per week in the span of just 4 weeks. This might have been the largest transformation in healthcare delivery, likely in at least in my lifetime. And it speaks to what might be happening for the future of clinical trials. So this great conceptualization of decentralized research studies is by Andy Korovos at a company that used to be called Electra Labs. And she highlights 2 dimensions to decentralized trials. The first is that where visits are conducted. Traditional research studies are conducted at research sites. And Andy says in the future, we're going to be bringing studies directly to participants. The second dimension is how the data are captured. Historically, data are captured through an intermediary, an investigator, someone like me, applying a research scale, whether that's the MBSU-PDRS, the NMSS, or the ALS functional rating scale, or many of the depression or cognitive scales. In the future, more and more health is going to be measured via mobile devices. These devices that we carry around with us all the time are supercomputers that are going to give us a wealth of data that's collected not in clinics, but in the real world. And obviously, in between these 2 extremes are going to be hybrid models that incorporate some elements of traditional research studies, some activities done at sites, some of it via intermediaries, but more and more will be brought directly to participants, and more will be captured via digital device. These decentralized studies aren't new. Craig Lipset, my apologies, and his colleagues at Pfizer conducted an entirely remote clinical trial 10 years ago, demonstrating the feasibility of this approach. And the COVID-19 pandemic disrupted our traditional clinical research studies, and it did so in a big and bad way. You can see the data on the number of trial suspensions, number of trials that were delayed or paused, were all enormous, and many studies were left scrambling for countermeasures to enable their studies to continue on. My colleagues and I here at the University of Rochester have been doing decentralized or virtual research studies or remote research studies since 2012. One of those studies is we're partnering with 23andMe, the direct-to-consumer genetics company, and we're following individuals who are genetic high risk for Parkinson's disease because they contain a mutation, because they carry a mutation in a gene called LARC2. And rather than setting up research sites throughout the country to follow these research participants, we're doing it all by video visits and by assessments that are done entirely remotely. We have 250 over 250 research participants from 35 states participating in the study conducted from a single site here at the University of Rochester. There's no in-person visits. There's only one IRB to deal with. And we do video visits like we're doing right now, and very similar to what you would do for clinical care. We do smell testing, we do standard cognitive assessments, assessments of mood, sleepiness, and other things that you would do in Parkinson's disease. And rather than that study being disrupted in the setting of a COVID-19 pandemic, we actually completed enrollment during the pandemic, and if anything, saw a slight acceleration in enrollment in that research study during the height of the pandemic. So that rather than being disrupted, this was a study that actually might have benefited from the disruption and variability that was occurring. For those of you who like to read Nicholas Taleb's book, Anti-Fragile, this would be an example of something that's anti-fragile that benefits from volatility. And the big takeaway from these decentralized research studies, much like telemedicine, is that individuals, participants, or patients love them. And this has been a consistent finding in all of our studies, and you can see satisfaction rates, well above 90% for virtually every aspect of the visit that we assess. And these traditional studies these new studies offer distinct differences from those of traditional. Recruitment expands from those activities done at sites to using registries or social media. Consent is done electronically. The study visit location moves from often expensive research sites to inexpensive sites like people's homes. We've done it in people's hotel rooms, workplaces, parking lots, even national parks. Study drug for some of those studies can be sent directly to patient's home. In fact, one study looking at hydroxychloroquine, whether it would work for post-exposure prophylaxis against COVID-19. Mailed study drug or matching placebo to hundreds of research participants throughout the country. Assessments move from those done in person to those that are done remote, either by video or capturing information through digital devices. In that study of the LARC2 carriers, we even sent Unified Pennsylvania Smell Identification Tests, or UPSIT, scratch and sniff test to have people do those assessments in the home. You can think that blood work and other things could be captured in the home. The burden on the participants, the financial cost to participating in research studies is much lower, and we try to get early and sustained involvement of participants in the studies. So just like the decentralized studies will change where studies are conducted, I think these studies are going to give us new insights into disease. And I think we're going to find people suffer a lot more than they let on when we have them in the clinic. So analogy is that when they're in the clinic, they're under the street light, but the vast majority of the time, 99.9% of the time that people are living, they're not in a research site. They're at home or carrying on their lives, and we're missing things like what portion of the day that they're alone, or what portion of the day that they're sedentary or lying down or sleeping. All these things which might be more meaningful than our traditional rating scales we entirely miss. And some of our traditional rating scales really just aren't designed to be administered remotely, suggesting that we're going to need new outcomes measures for the disease. We've done some early work with smartphones showing that on the left, that individuals who take levodopa, the most effective medication for Parkinson's disease, have clear benefit in response to taking the medicine, with improvement in their mobile Parkinson's disease score, with lower scores being better. And you can see that happening over 6 months. By contrast, on the right, we have 3 data points from a traditional research study in which you might do assessments at baseline, month 3, and month 6. By comparison, we had 150 plus assessments of the smartphone in the real world done by research participants. These assessments can be extended to the home, and rather than using portable devices or wearable devices, you can use even invisible devices My colleague, Dr. Dina Katabi, and her team at MIT had developed a device called Emerald, which is a radio wave sensor. It looks like a Wi-Fi router and emits radio waves that reflect off individuals and then detect those reflections off things that carry water. And you can see how much time this individual spends in their bedroom, how much time they spend in the bathroom, unfortunately a lot of time, where their favorite chair is, and even the path by which that they walk. If you look at these concentric circles, each concentric circle is a 24-hour period, and this is for 6 individuals, starting at midnight or 0 and going to 12 midnight and then into the night. And you can see the time that they're in each. There were 50. This is an 8-week study, so there are 56 concentric circles per person, and you can see that some of the individuals clearly have a circadian pattern to their time in bed. They're going to bed at 9:30 and getting up at 4 or 5 in the morning, whereas others have complete loss of their circadian pattern. And some individuals, participants 5 and 6, are spending essentially half their day in bed, unlikely to be a sign of good health. I'll conclude that we've just seen huge transformations in how we deliver care, and I think these transformations are going to extend to clinical research. Vladimir Lenin, likely speaking about the Russian Revolution in 1917, said there are decades where nothing happens, and there are weeks where decades happen. And I think we've experienced those weeks, and we can take advantage of those weeks that transform clinical care and clinical research to think about how we can design clinical trials in the future and bring trials to participants instead of participants to trials. Thanks very much.
Hello, I'm Dr. Laura Lee Johnson, one of the division directors in the Office of Biostatistics in the Center for Drug Evaluation and Research. People think about developing treatment or prevention for COVID-19, but FDA has also worked on guidances on how to help trials where studies or patients enrolled in trials are impacted by COVID-19, including public health emergency measures such as quarantines or considerations, such as if site personnel or trial participants become infected with COVID-19. We're now closing in on almost 40 pages of questions and answers available online, drawn from many of the questions we've received and lessons learned. These topics include a variety of things like remote assessment, a variety of ways to satisfy FDA requirements for obtaining and documenting informed consent, using alternative laboratories or imaging centers, and other issues that have arisen. Many times, the world regulators like FDA are not the stumbling blocks, however. We're trying to keep a focus on ensuring the safety of trial participants and study staff. We then think about the trial integrity and how data can be compiled and analyzed at the end of a study when decisions need to be made about if a medical product should or should not be authorized or approved. To this end, we have a statistical considerations guidance for trials, since changes to trial conduct, how primary and secondary endpoints or safety information is collected in a trial, how missing data or data outside of previously defined windows can be used, how all of this may impact the analyses and interpretation of trials. How this is handled in your statistical analysis plan is going to take very careful thought and planning. As we learn more, we update our guidances, and I encourage everyone to look at FDA Guidance's website for the most recent updates. Once you've taken human safety into account, and here I mean for participants and personnel, collecting information about what's going on in the context of a study and the patients in it is vital. Did you go from collecting a 6-minute walk test in person at your clinical site? Now you're trying to collect it in patients' living areas so they don't need to come in? Recording how data was collected, where it was collected, instructions given, issues that came up, and if it's missing and why it's missing is vital. We've worked with CDISC, so data standards were updated for ongoing studies disrupted by COVID-19. So the data structures collected what we call metadata for trials and studies. In these cases, you don't just have nothing if a visit is missed, but information on why. And this will help with the eventual trial interpretation. This is especially important in rare disease settings. Why? Because you see a lot of data reuse, and I want to focus here for a minute. 10 or even 20 years from now, when you or others are looking at natural history data, randomized trial data may be from a control arm or from the treatment arm that was eventually approved. These are the types of data that are going to get borrowed to help determine how to design a future trial, maybe to augment a control arm for a trial, maybe to set a non-inferiority bound. And knowing what happened from January 2020 through the pandemic will help decide the parameters for simulations that your statisticians are going to be running to help do all of those things. Maybe to help determine whether this study should be part of a meta-analysis. Was the impact of COVID so large that it's a reason not to include data in any of these settings or to weight it differently? This is an important part, as sometimes we talk about cherry picking, but here you might have some real reasons for data exclusion or for reinterpretation. So that's why this metadata, the whys, are really important for trials and studies that are underway now and how that data may be used in the future. One thing that gets hidden many times is that data borrowing is an important underpinning for many studies, even if the data is only used to help design the next study. If you have any questions about remote assessment, especially for clinical outcome assessments of any type, I encourage you to look at our guidances. I'll also note that not all technology is robust or fit for purpose for use in clinical research, as it might be for your own personal data use. Not all technology stores the information needed to recreate the summary information, and the validity and reliability when you start to look across people and across situations and over time might be shockingly poor. And also, not everyone has the technology. Health equity has been an issue we've pushed when trials talk about making changes. Most folks that are watching this probably have a smartphone or easy computer or internet access, minutes to text, and they can use their phone in a variety of different ways. What gets forgotten is too often, many people in the United States may have been standing outside a closed library to get free Wi-Fi to fill out your survey because they didn't have minutes left or they lacked money to buy more. Their kids were trying to school at home and they literally no longer had time or bandwidth. They may not have regular electricity or internet access here or around the world. When we're thinking of solutions, we challenge researchers to think not only who are they including, but who are they going to be leaving out and burdening? How does that impact your findings? What are the ways to have representativeness and useful data? As we take the lessons learned and think about ways to change trials for the future, this too is a vital part of our discussion, especially in rare diseases where reducing the need for patients to gather at a few centers around the world could bring important access to trials and studies for many families. With that, I'll conclude. As we get more questions, more trials done during this time come into regulators and we learn more potential issues and also innovative and useful solutions, we will continue to update our advice. Thank you very much for your participation in this important endeavor.
Hello, everyone. I'm Ann Pariser. I'm the Director of the Office of Rare Diseases Research, or ORDR, at the National Center for Advancing Translational Sciences, or NCATS, at NIH. And I'm going to be talking about clinical solutions following COVID for our panel today. So just by way of a very brief introduction, I am from a different center, NCATS, at NIH, and many thanks to NINDS for inviting me today. And at NCATS, just briefly, we are one of the 27 NIH institutes or centers, and we are actually the youngest and one of the smallest. So we were established about 10 years ago in 2012. And we are the only center at NIH that is focused on translational sciences. So by translational sciences, we mean the process of turning observations, such as discoveries in the lab or in the clinic, into interventions that will improve people's health. So this is a graphical display of the same thing, but what we're trying to do here in the upper left-hand corner is what we're trying to do is get more treatments to more people more quickly. So the problem is we have thousands of diseases, most of them are rare diseases, but only hundreds of treatments. Only about 5% of diseases have an FDA-approved treatment. And it still takes a long time to develop a therapy. On average, it's estimated about 10 to 15 years. And 95% of the time they fail, and they fail for a variety of reasons. But that's what translational science is all about. We're trying to improve that process to try to get more of these treatments through in less time, and develop things like predictive models, better clinical trial design, or better understanding of diseases that can help multiple diseases at a time. And my office, the Office of Rare Diseases Research, is specifically focused, as the name implies, on advancing rare diseases research to benefit patients. And we do that through a variety of programs, but we don't focus on any one particular disease. We're trying to help all the diseases and rare diseases research process generally. So that is just a little bit of background. Today's topic is on clinical solutions following COVID. So got a graphic here of a novel. Just apologies to Gabriela Garcia Marquez, although the persistence and the passion shown by the characters, I think, is very reminiscent of the current pandemic situation. But just a few highlights for background for the panel is that we conducted a COVID-19 patient impact survey, and I'll tell you a little bit about that. And that led to some additional insight that helped us develop some new NCATS initiatives. So this COVID-19 patient impact survey, so this was actually performed last year in 2020. During the pandemic, and it was stood up very quickly by the Rare Diseases Clinical Research Network, or the RDCRN. So what is the RDCRN? So very briefly, it is a network of centers of excellence for rare diseases, and it was established about 18 years ago. And these are 5-year award cycles, and we're now in the middle of the 4th award cohort. And the purpose of the RDCRN is to try to facilitate rare disease research through the establishment of these research consortia. And this is a little bit unique in that the consortia have to study at least 3 rare diseases or conditions within the consortium at the same time. So it's a many diseases at a time approach. And patients, researchers, clinicians within these consortia, they all work together, including patients as leaders. And 2 of our big priorities are things like clinical trial readiness and data sharing and data standards. But it was because of the framework of this network and this broad participation, we're able to cover about 250 different rare diseases within the RDCRN and over 100 centers and over about 140 patient groups. We were able to actually stand up this impact survey very quickly when the situation arose. So this is just a graphical representation of the RDCRN. So there are 20 consortia that collaborate together. And just a quick look here, you can see that they're very, very diverse. And this is a collaboration across NIH, researchers, patient advocacy groups, but also across 10 NIH institutes and offices, including NINDS, who is actually our biggest partner here. So it's a really strong collaboration, and it did provide this platform or this framework where we were able to conduct the survey. So I put a link here if you'd like to learn a little bit more about the network. So about the survey, so this was initiated about June or July or so of last year by the Data Management and Coordinating Center, or the DMCC, and several of the consortia who all worked together to put the survey together. So this was a cross-sectional web-based survey, meaning that it was a one-time survey of rare disease patients or their primary caregivers. So you could be a patient or a primary caregiver, and you had to be based in the US. So this was actually open to the entire rare disease community. It did not have to be part of an RDCRN, but the RDCRN centers, as well as the patient groups, did help disseminate and increase awareness of the survey. So what did the survey seek to better understand? Well, we were trying to understand the impact of COVID on the rare disease community. So there were a number of questions, but some of them were things like access to treatment and care during the pandemic. Who is becoming infected? Is the clinical course similar to what we're seeing across the general population, or was it different? And what kind of stressors and impacts did it have among patients and families, among many others? So some of these questions actually came out of what we were hearing from the patient community about their concerns about many of these things. So the survey was open for about 6 months or so, and there were over 4,000 people who completed surveys. And we got responses actually from all 50 states, Washington, D.C., and Puerto Rico, and included about 200 or so different rare diseases. And for the people who participated in this survey, they then had the option to further participate in the NIH Sero survey that contributed lab samples that were then studied for things like COVID antibodies. And here's a link here at the bottom to the survey. So some high-level results. So there are patients with about 150 or so different rare diseases. About 65% of the participants were female, and about 90% were white. And about 17% or so of patients reported a diagnosis of COVID, and they also reported similar rates of hospitalization, types of symptoms, manifestations as other people with COVID. One exception here, though, there was about 44% of people reported that the rare disease somehow complicated their illness. And what also came through very strongly in the survey was that the pandemic did really negatively impact the rare disease community in terms of things like people reported difficulty with access to care, to treatments for the rare diseases, in some cases access to diet or specialized treatments or hospitalizations. So the rare disease community does tend to have severe or serious illness and are likely disproportionately dependent on things like emergency rooms and hospitalizations. So I guess this is not surprising that when the medical system was so stressed that these sort of issues would come to the forefront. And the community also did share changes in mood, anxiety, and increased stress with the pandemic. So the preliminary results were actually presented this year at Rare Disease Day at NIH, and put here the link to the video cast if you would like to see it. This was presented in more detail by two of our investigators, Dr. Rothenberg and Dr. Ferkel. And they went into more detail about some of the things that I've shared with you. But if you access the link, and at about 2 hours, 4 minutes and 9 seconds is when it starts. The results were also presented in a Spotlight on Rare Diseases newsletter. Here's the link right here. And the data is still being analyzed. So we expect to see more publications and more things coming out of the survey results. So what were some of the lessons that we learned? Some of the things that came out of this is we did see some impact on ongoing studies. So early on, there were some studies that were paused, but also there were problems with getting patients to treatment centers. So for example, some of the natural history studies noticed a drop off. For the most part, we think that we're back up and running, but we'll have to see how this impacts the future. What we also heard really loudly and clearly, though, were difficulties with remote capture. And this has long been an issue for rare diseases. Many of our patients have difficulty with getting to study sites. Sometimes these can be very geographically dispersed, or it may only be 1 or 2 or just a few sites that may be located far from where people live, or people who have mobility considerations, for example, or young children with health needs. This has been a longstanding issue. And we've known for a long time that we do need better remote capture, but I think this really came out during the pandemic. And the equity, diversity, inclusion issues, I think if you recall, I just said about 90% of our participants were white. And I think it was pretty clear we're not reaching all the people that we want to reach through the methods that we're using. So just a couple of things that this has resulted in is NCATS has presented a new concept in January. So this is usually the first step in getting, for example, a funding announcement is we present this to our council, which is like our oversight board. So what this is specifically for is we're proposing trying to develop new methods and validate remote measures for use in clinical trials for people with rare diseases. So this will be under a small business innovation research contact topic. So you check back, watch this space, and we will let you know as soon as something is publicly available. And this also builds on a prior concept that we have put out for augmented reality. And so this has a complicated name, but essentially this is trying to develop a virtual reality platform, sort of like a video game, if you will, that is able to capture high quality data, facilitate communication, customize some of these outcomes, or even perform teletherapy remotely. So this is something that also has been advertised, and should be more about that should be coming soon. So those were just a couple of things, and I'd be happy to answer questions. Thank you.
Okay, so now we are going to segue into our live question and answer. So I would love to ask all of our panelists and our moderator, Geraldine Bliss, to pop on. Get your videos going, get yourself unmuted. And Greg, Geraldine is having some trouble with her unmuting. If you could help her out, that would be helpful.
I'm good now. Thank you.
Oh, we got it. All right. I'll let you take it away, Geraldine.
Yeah. So thank you to our panelists. We appreciated your talks delivered remotely. So we already have a question from Ron Bartek, and we're going to start with that one because we have a hard stop at 12:30. Dr. Dorsey, thank you so much for your insights regarding the future of clinical trials. You spoke of developments in remote collection of data for clinical outcome measures. You illustrated with an example of the technology to monitor such things as sleep time. How do you believe development is going in regards to technologies to measure accurately, analyze, and report to regulators the patient's ability to perform their activities of daily living? How long do you think it will take before such technologies are in regular effective use in reporting clinical outcomes in all clinical trials, remote and in clinic?
So thank you very much for the question. Can you hear me okay, Geraldine?
Perfect.
Perfect. The technologies are around us everywhere we go. I mean, we have supercomputers that we carry around us wherever we go. Some of us wear supercomputers. Some of us have our homes outfitted with supercomputers. So the technology is not the issue. The issue, I think, is to the second part of the question. Are we going to use these technologies, which can provide us real-world, objective, frequent, sensitive assessments of health or disease in our clinical trials and clinical research efforts? And I think the answer to that is increasingly yes. We heard a little bit from NCATS. I think we heard a little bit from the FDA. We heard an experience from the investigator and did their day-to-day experience. And they're generally increasingly open to doing this. If you look at a database controlled by the Digital Medicine Society, DIME Society, it lists all the digital endpoints that are being used in clinical trials. And that list is increasingly long. The FDA has accepted moderate to vigorous physical activity as an endpoint in trials in pulmonary fibrosis. The EMA has accepted measures of gait velocity as an endpoint in Duchenne's muscular dystrophy, which is apropos to this audience. So I think we're going to see these things increasingly used and adopted. The sooner regulators and the sooner pharmaceutical and sponsors and researchers incorporate these things into trials, the sooner we'll see their use, and the sooner we'll get therapies for a lot of the diseases for which we lack highly effective treatments.
One of the things that I think almost all of the speakers touched upon very briefly was the opportunity to increase access to clinical trials and clinical research studies to participants who might not traditionally be able to participate. And in neurological conditions, it can be because of comorbid behavioral issues, mental health conditions, motor issues. These may all be barriers, but also there have been issues with sort of like cost of taking time off to participate in a study, or trying to cover the travel expenses of participating in a research study. And we heard a little bit about that in Dr. Johnson's talk. I'm wondering if Dr. Dorsey and Dr. Barry Kravis, if you've yet seen any sort of change in the representation of participants in your clinical trials and clinical research studies through the remote visits or remote trials.
Yeah, I think we're beginning to see some of that. We have a trial going on in a neurodevelopmental disability that is recruiting, and I think most of our patients came from the Chicago area or Illinois or maybe southern Wisconsin or Michigan, but now we're starting to see people come in from Kansas and Oklahoma, and because they only have to come to our site a limited number of times, and we can do a lot of the visits remotely. And there really are a lot of neurological scales that you can capture most of the important things remotely, and then get the key things that you can't capture remotely when you actually have the patient there in clinic. And I was very skeptical about this at the beginning, and I honestly was thinking, okay, it's gonna be impossible for us to collect some of these outcomes, because we're not studying just a motor measure or a gait velocity. We're trying to assess language and cognitive things and functional things, and we didn't have in these trials really anything digital to capture that. But I've been actually surprised at how well video conferencing has actually captured it, and how well we've been able to instruct families to carry out specific steps of the exam as if they were the examiner, and kind of examine the patient through the family. And I think a lot more thinking to kind of standardize that process is going to be needed. But I think this forced us to really think hard about how to do that. And particularly in our NeuroNext study with our SLPs, the way they figured out how to deliver a language assessment remotely through the families. And we have data before and after that kind of sandwich all the remote assessments. So we're going to be able to very carefully look at whether this assessment is the same in person or remote, and really start to validate measures like that, which I think are going to help tremendously in terms of rolling out studies, addressing things like cognition and language.
I would just say briefly in our experience that we've addressed geographical barriers to participation. We have people participating from all different parts of the country. I think some of the social barriers to participation and the lack of trustworthiness among researchers like me and academic institutions is a significant barrier to others participating in these studies and in studies in general.
I've received a great question from Janet Heischetter. Do we see consent being done virtually going forward?
We're doing it all the time, so.
Yeah, I agree. I mean, there's a whole method in REDCap for doing consent, and you just get it. You just say you're going to do that process with your IRB now, and it gets approved very easily. I've been on the IRB for the past almost 20 years now at Rush, and there was a lot of resistance to this, say 10, 15 years ago, but now it's considered almost standard.
And I was really delighted to hear in your talk that the IRB had been so flexible and, modifying protocols as needed because of the pandemic. I am kind of wondering for Dr. Dorsey, you talked about the use of the Emerald device. I mean, I think there are always special considerations about privacy when people are utilizing wearables and neurables like the Emerald. So were there any special concerns about privacy that you needed to address with the IRB or with the regulators?
Yes, and we survey in many of our studies, we survey participants about their concerns about privacy. I think people with disease have different views of privacy than people without the disease. But I think these are very real concerns, and I think we're going to need more privacy protections in place, just like we have the Genetic Information Non-Discrimination Act, which came in 2008 to protect genetic information. I think we're going to need a similar set of laws to protect the privacy of health and other related information going forward.
Right, I think that's a really important topic. Great, we have another question that came from Heidi Grabenstatter. Dr. Barry Kravis did a lovely job at emphasizing the importance of SLPs and development of communication measures, and the Rush SLPs helped develop tools for others for remote methods. Can she comment on the need to include their voices at the table with regulators, given they are in the weeds with the patients and caregivers? That is, they may have more touch points than a clinician would.
Okay, yes, I think that the SLPs are important in this process. I think the process in general is that we're trying to develop more individualized outcome measures and validate language measures that cross multiple levels of function. And that has been a bit challenging because these aren't like the standardized measures that have years and years of standardization. But I think one of the really important points that the SLPs make to us all the time is that the standard measures are not made for people with intellectual disability or severe language delays. And so even though the standard measures are validated, they're not actually validated for the patient population that we're studying. And so some of the new and novel measures are much more sensitive at picking up true changes that are life relevant in people with intellectual disabilities and language disorders. And so we will likely need help from the SLP field in general to help us make these measures be measures that can be used for by regulators for treatments that are being developed for people with developmental disabilities.
So we have like a little over 1 minute left. Dr. Barry Kravis, you mentioned that drug mailing and bottle return turned out to be one of the bigger challenges. I would like for both Dr. Dorsey and Dr. Barry Kravis to sort of, if you can boil it down in a nutshell, what is the one major challenge you think in general is going to be a barrier that disease groups are going to have to figure out in order to conduct successful remote or distributed clinical trials?
I'll go quickly. Most people want to participate in research. It's incumbent on us to enable people to participate in research on their terms and instead of our terms. And by bringing studies directly to participants, this is a huge way of increasing access to participating in research, which vast majority of people with diseases want to do.
Right.
I agree. It's incredibly important to try to bring the research to everyone. And I think the remote process helped us do it. I will say that we're relatively early in this, and it's not easy to get drug and to get labs and to get things that usually would have required the patient to be physically on site done at the patient's house and done well, and you wind up having repeats of things. And I think we'll get better at this as time goes on. We were just like thrust into it instantly with a whole bunch of studies all at once, and it was a lot for all the coordinators to keep track of. But I honestly think that we need to try to make it easier because it is so important, particularly for a rare disease population where most of your patients are flying in from somewhere in the country.
Great. Well, we're excited about this new type of clinical trial, and we're all hoping this will be successful. I really see it bringing a lot of benefit to patients with neurological disorders. So thank you to all of our panelists. I'm sorry, I wish we had a little more time for questions, but I think we need to wrap it up. And I believe we're all going to be routed. If we've signed up for the one-to-one sessions, we're automatically going to be routed into those sessions. Otherwise, I think we just hang out here and chat. Sam, do you have anything to add to that?
That was perfect, Geraldine. Yes. So we are going to shortly be routed. If you did sign up for our one-to-one, you are going to end up in a teeny tiny breakout room with the person who you requested to speak to. The rest of us will hang out here in this central meeting space. You're welcome to take a break, but you're also welcome to continue to chat. And I do think that, Geraldine and the panelists, that was a fantastic session. And there is some time for us to continue to talk a little bit in here as a group. But with that, I actually am going to ask our logistics contractors to go ahead and get the people who did request to have these specific conversations with our NINDS program staff into the appropriate rooms so we don't take up any of their time.